Yep.
This is like really high. Oh, it's like adjustable. That's a bit cool.
Oh, it's adjustable.
Mm.
Oh, cool. I did not know that. I would have been. I've been hiding behind, like, peering out of the top.
Yeah. It's better lower, I reckon. Yeah, so you're not climbing. I want one of those for my desk. That would be cool. Adjustable desk.
Melissa's got one.
Oh, does she?
She's put it on top of her desk.
Ah, like an on-top thing. Right. Oh, that's really correct.
Yeah, go on the crowd.
Here we go.
Cool. So I just wanted to make a couple of quick announcements before.
Sure.
Just actually, might have been just one actually. One or two, yeah. Yeah. 1 minute.
Yeah.
Cool.
Oh. I'm going to steal that from you.
Yeah, no worries.
So I have a soft voice. I'm not so good
It's
at
not
the whole
recorded
projecting.
otherwise.
Yeah. This one sometimes does something, but oh, yeah. It's like a pretty, it's got a bit of a load on top of it. It's
It's
special
pretty decent.
lectern. Oh,
Pretty decent stage around.
yeah. It's quite smooth too.
It's hydraulic. Or
I don't know,
Sound
what do you reckon?
like a bit screwed or something.
I reckon.
Thanks again for doing this. It's good. I think they've enjoyed the last. Had two previous guest lectures.
Okay,
It's always good.
mixes it up.
Yeah, next week's the guest lecture too. So.
Yeah, wow. It's a great way to do it for you.
Yeah, well, this is a legacy of other lecturers doing it like that, and then,
Yeah.
yeah, it works out pretty well, I think.
Yeah, if that's like,
get the
if it
advantage.
suits the course, then
Well, it's
yeah.
a final year elective,
Exactly.
so it's sort of you get
Yeah.
the advanced topics.
Because some of the subjects I normally lecture, like first year radiography, like they need to learn like what
Yeah.
this is and that is before they can do the rest of the course.
Yeah,
So
yeah,
it's
yeah.
like,
Sort of have interest.
yeah.
Which is good.
They're usually not that excited to learn it.
Yeah.
They're like, physics. I didn't choose physics. I chose real.
Right.
Yeah.
Yeah, to get started.
Yep.
Do it.
You should change the
Yeah,
light.
you can do that. I always get this wrong. Room settings. Hello. Cool. Hi everyone. Thanks for coming to this guest lecture. I just had a couple of quick announcements I wanted to make beforehand. Firstly, the SETU evaluations are now open, I believe, or will be soon. If you can do them, that would be really awesome. It gives us feedback on the course. We've changed the course a little bit this year, so it's really great to get your feedback on that. And the more people that actually do it, the better the feedback is. So, yeah, really encourage you to do that, especially if you've liked the course. Put that in your numerical scores, and if you have any sort of constructive criticisms, you put that in the comments. That's fantastic. Also, the second thing is that the second quiz will be open on Thursday. I sent an email if you didn't catch it. Yeah, so it'll be the same format as the previous quiz. It's on this Thursday, and it's on the 3 lectures that I delivered in weeks 5 to 7. Yep, and that's all for me for now. So let's introduce Dr. Kay Morgan, who has kindly agreed to give a guest lecture today. Kay's hugely experienced at synchrotron imaging. She's recently was awarded the ARC Future Fellow, which is like the most prestigious and competitive fellowships that the Australian government gives. She's been to all the synchrotrons around the world and done experiments there. Spent a fair amount of time in one of the leading X-ray labs in Germany, and is back here now and is going to speak about X-ray imaging on synchrotrons. which is a very specific kind of imaging, and particularly phase contrast imaging, which is different to what I spoke about, which is absorption contrast imaging. So I'll hand it over to Kay, and thanks very much.
Thanks, Steve. Thank you for having me here today. So I'm going to kind of go through a few different topics, a bit about imaging, a bit about synchrotrons, and then finish up with some applications of how we're actually using the synchrotron to make a difference. So X-ray imaging, you're probably quite familiar. Steve has talked to you about attenuation-based imaging. So back from Röntgen's image in 1985, we're now getting much higher resolution images. We're looking at smaller objects, and we're also looking at them at high speed. And so I'm going to show you some of the techniques that we've developed to be able to do that. So why would you want to image with X-rays? There's lots of other possibilities. The first thing is they're non-invasive, so you can see through objects. So something like electron microscopy gives you higher spatial resolution than X-rays, so you can see much smaller objects, but it can only ever give you the surface. You can't see more than one or two layers of atoms. So X-rays can see right through. They're high spatial resolution, so we can see really small objects, less than a micron, as you can see here, so a single cell organism. We can take images very quickly, so we can see things that are moving, so this liquid sloshing around inside a ventilator thing. So something like MRI takes several seconds to capture an image, so there's no way you could collect something like this with MRI. And also you can access 3D using computed tomography. Did Steve talk to you guys about that at all? Maybe? No? So if you take images at all different angles, then you can use some maths to reconstruct a 3D volume. And so these examples here also kind of give you a scope of the main applications. So security, research into the world around us in terms of these single cell organisms, industry developing new products like the ventilator thing, and then also medical, obviously. So in this talk I'm going to go a bit from the conventional X-ray imaging to what we call phase contrast imaging. So conventional X-ray imaging is basically a shadow image. We're basically looking at where is it that most of the light isn't getting through anymore, there must be something dense there stopping it. And of course we usually see an X-ray image kind of inverted from what we'd see from a shadow. So the shadow here is black and the bone here is white, and that's because we've used film conventionally for X-ray imaging, which always gives you that inverted image. So what about if we've got something that we want to image which doesn't cast a strong shadow? So something like bone gives you a strong shadow, but something that's made out of the kind of softer tissues that are around the body, so muscle or fat or skin, the lungs, the brain, they don't cast a strong shadow. So we typically don't see much if we take an image of one of those things. The X-rays just go straight through. So by visible light analogy, if we had a glass of milk, that would give you a nice strong shadow. The light doesn't make it through. But a glass of water, the light makes it through, and so it's quite difficult to see it. So we can talk about strongly attenuating objects, create a strong shadow, weakly attenuating, not so much. So the question that I try to answer in my research is there a way that we can use X-ray imaging to still see those weakly attenuating structures like the soft tissue, so the fat and the muscle and the skin and the organs that are soft? So in order to do that, we have to look at the other properties of the X-ray wave field instead of just looking at does the X-ray photon make it through the object. We want to look at the X-ray wave field and say how is it changed by the object? What other properties are there in there we can use to extract information about the object that X-ray wave field passed through? So if we're talking about light, as we can describe it as a wave, so it has an intensity which corresponds to the amplitude of that wave. And it also has a phase difference. So if we look at the blue and the black waves here, there's a difference where the peaks lie. So they don't line up perfectly in this image. We'd have to kind of slide across like that for them to line up. So we see what we would call a phase difference between those waves. And with X-rays, we can measure the intensity. That's easy. It'll tell us how bright the light is, how many photons made it through the sample. But we don't have any way to measure phase. There's no kind of detector or camera that can just tell you the phase of this wave is this compared to another one. So there's no way to directly measure it. So the thing that's quite useful though is if we can get that phase information, we would be able to see these soft tissues. So we can describe a particular material, whether it's tissue or whether it's some manufactured material, anything, using its complex refractive index, which is this n equals 1 minus delta plus I beta. And so it describes how likely the X-ray wave field is to interact with that material in different ways. So the first way is absorption. So you can see a wave going through that pink material and it gets through kind of unscathed, the same intensity, the same phase. But if we look at the wave that's gone through the white material there with a different beta value, we can see that it's reduced in amplitude, so there's not as much light getting through. And if we do that, we can get an image of bone, for example. So this is the head of a mouse. And we've got quite a low energy X-ray, so even the soft tissue surrounding the bone is blocking some of the light. The other part of that complex refractive index is the delta. Sorry, I should say also that I there is like the square root of -1, so it's probably J over here in engineering, I think. So if we're talking about that delta value, that's going to determine the phase changes as the wave field goes through some particular material. So if you compare the waves that are going into the pink and the white here on the right, you can see that they go in where the peaks and the waves are lined up, so they're in phase with each other. But then when you see they've come out of the material, suddenly they're not lined up. So the white one has its peaks where those vertical black lines are, and the pink one has the peaks where those pink lines are. And so there's suddenly this phase difference. And so we'll see that occur if we have two different materials that have two different delta values. We can also kind of connect up the peaks of those waves like this, which kind of gives you what the wave fronts would look like. So when we're talking about a change in the phase of the X-ray wave field, it's also kind of like a refraction in that the wave's coming in at one angle and then it's going out at a different angle. So it's coming straight across here, but then when it goes out through the material, it's kind of heading a little bit down at an angle as well. So if we can use this information, then we can see a lot more structures. So here you can see with some extra phase contrast in the image, you can see all the hairs and the fur that are on the outside of the mouse's head. You can see the little tube that he's resting on. You can see his nasal airways there. And even if you look kind of in the middle of the image here, you can kind of see all that there's extra definition in there, and that's all those the airways that go from your nose down towards the back of your throat, which are in there, and you're getting a lot more contrast from those. And that's because there's a big difference in that delta value between tissue and air. So we can get a lot of contrast between tissue and air. So if there's ever any part in the body where we've got some air, that shows up really well. We can also combine this. This is a single projection image. If we take images at many angles, we can get a computed tomography slice, and if we have the phase contrast information in there, then we can see all the airways. So this is slicing through that area there, and you can see these are all the kind of curly airways in the nose that are there to catch any dust when the mouse breathes in. Then there's kind of the bone around his nose, and then his jaw kind of coming down here. There's a tooth you can see here and here. Okay, but the question is, how do we get that image? How do we measure this phase? And so I said there wasn't any kind of detector that can capture phase information. So what we need to do is actually convert the variations in the phase into a variation in intensity using some kind of optical system, because then we can measure it. We can measure intensity, so we just need to somehow convert phase into intensity. So a very simple way of converting phase into intensity is a lens. So the lens focuses the light, so it introduces these phase variations so that the waves are all kind of heading to the same point. If you were to trace some rays, you can see that they're all heading to one point, and if you place a piece of paper there, you see a lot of light is landing in the middle. So we've got what was originally variations in phase right after the lens, now results in variations in intensity at this piece of paper here. So we've got variations in phase becoming variations in intensity. If you look at a magnifying glass, you can see this quite clearly. We've got this nice bright spot in the middle, and then it's kind of dark where the intensity has kind of been taken from to concentrate it in the middle. So that's, it is possible to do this conversion. So the easiest way to do this with X-rays is really this same kind of setup here, where we're just introducing some distance between the phase object, we would call it, something that changes the phase of the wave field, and the camera system, whether it's a piece of paper or an X-ray detector. So having this distance in here allows the X-rays to kind of add together in some areas, disappear from other areas, so that we've got variations in intensity. So if we imagine some kind of object here, this pink object here, and we've got the wave field coming in here, if we were to plot the phase, you'd see kind of a phase shadow behind that, and there's a different phase behind the object as there is on the sides of it. If we plot the wave fronts here, you can kind of see that. And if we imagine the wave that's gone above the object here, it's still heading straight across, whereas the wave field that's come through the object is now kind of heading up at an angle as well. So you're going to have the wave field that's gone above running into the wave field that's kind of coming up here, and when they run into each other, they're going to interfere. When they interfere, we end up with these bright dark fringes in the intensity. So as kind of what's really happening is we've just got constructive and destructive interference. So you're probably familiar with the two-slit pattern, where we've got two sources of waves here. It's two balls bouncing on the water, but we can imagine that it's creating these light waves. And we get places where we've got constructive interference here, where the waves are in phase and adding together. And then we have lines of destructive interference, where they're completely out of phase, and so when you add them together, you get zero amplitude along here. And so that's effectively what we're seeing here. We've got constructive interference. When they're in phase, they're adding up to create brighter light, which is shown in the white here. And then there's some places where they are completely out of phase, so when they add together, we get this lowered intensity, this black stripe coming out here. And so we call this pattern a fringe. So we get this kind of bright dark fringe from any edges that we have in our object. So we'd stick the detector at one kind of plane through here, and we'd see those edges. And so we can see that here with some images of the airway, where we're simply moving the camera back further and further, that those edges of the airway, so the airway's in the middle here, become thicker and more easy to see with more and more propagation. And so you can kind of tune exactly how much contrast you want just by changing that propagation distance. So whenever we're doing this, we need to have sufficiently coherent light. So this isn't always possible at any kind of X-ray source that you might use, and so we typically would use a synchrotron. So a synchrotron is a big research facility that produces very, very bright light. These are a couple of the synchrotrons that I've done work at. We've got the Swiss light source near Zurich up the top, Australian synchrotron, which is just across the road, and then in Japan, where I've done most of the experiments. So this one, they actually had trouble finding a flat enough surface to build the synchrotron, so they built it around this mountain. And so you can see there's like a car there, so it's quite big all the way around. So in the synchrotron, they accelerate electrons to 99.999% the speed of light, so it goes around this mountain around 1 million times every second. And as it goes around that loop, it goes through different devices that mean the electron will oscillate or turn a corner, and every time it does that, it will give off very bright light. And so there's a whole lot of experiments. Every place where there's one of those arrows, we've got an experiment taking place and some light coming out of the synchrotron. So the Australian synchrotron is one of the most compact designs, and they actually accelerate the electrons within the main storage ring. So they come out of this electron gun, they're brought up to speed using the linear accelerator and the booster ring, and once they're at the right speed, they're injected into this storage ring. And then we have, at each of these points here, we've got a blue arrow. The electron goes through some kind of magnetic device, which means that we get bright light coming out to an experimental station. So there's three main devices that are used to convert, to make the electrons move in such a way to produce light. So we've got a bending magnet where they just kind of bend, and if you've got an electron changing direction, it will give off some type of electromagnetic radiation. And then the other ones we have is an undulator or wiggler, which looks like in the bottom slide here. And so this is basically the electrons passing through at high speed. It goes through a north-south. Using your right-hand rule, you can see that the force is going to be one direction. And then when we switch to the north-south, it's suddenly in the opposite direction, so it pushes that electron back and forth. So it's kind of like having a current in an antenna, which is a whole lot of charges moving in the antenna, gives off radio waves, for example. But here it's much, much smaller oscillations and it's giving off X-rays. And so there is, at the synchrotron, there are some other wavelengths you can get, so infrared, for example. But most of the beamlines, you've got X-rays being produced using either an undulator or a wiggler. So the difference between these is just how small the period is between the magnets. So the wiggler has a smaller period of magnets. So all this is controlled by some guys in a control room. This is the one in Japan. So it's quite nice because we don't have to worry about keeping the machine running. So this is inside the synchrotron in Japan. It's quite a long ride around to the experimental station that we use, and you have to kind of go over cables, and at some points you have to go underneath the beam lines that are carrying the X-rays to the experimental stations. So like there. And you can see they've really packed in a lot of equipment. And it's kind of scary that they allow us to ride our bicycles through. This is like multi-million-dollar equipment either side of you, squeezing through. So there's kind of a quick tour of the synchrotron. So once we get to our experimental station, it looks something like this. This is kind of standing in the doorway, and there's this giant lead wall which separates the X-rays and the experiment from the scientists. So we are never going to be in that X-ray hutch when the X-rays are on. So we call this big metal room a hutch. I don't know why, it's kind of a strange word, but with this door would always close before the X-rays are allowed to turn on, and we can then control everything from out here at the computers. So we can move these motorised stages to move the camera and the sample and the optics to the right positions, and we can monitor everything from outside while we're doing the imaging. A couple more images here. This is inside the Swiss synchrotron. They've got a big wooden roof. This is one of the, they have these kind of optics conditioning hutches that are up near close to the source, where you might use, for example, a crystal to bounce the light off so that you get rid of all of the wavelengths except for the exact wavelength you want, called a monochromator. This is inside the hutch, so some of the experimental setup, and this is the hutch at the Australian synchrotron, which is much, much bigger than the one in Japan. And so the plan for this hutch is actually to do human imaging in the next few years, and also radiation treatment there. So it produces actually the biggest synchrotron X-ray beam in the world, which you can see over here. This yellow paper turns red when it's exposed to X-rays, so that's the X-ray beam there. So it's a bit of a contrast to this beam in Japan is around 1 millimetre by 1 millimetre, the field of view, so much smaller. Okay, so this all looks very elaborate. Why do we need to use the synchrotron to do this imaging? So the first thing is the synchrotron produces very, very bright light. So that allows us to take very short exposures, and so we can image things that are moving really quickly and still get enough photons to create a nice image. So if we had very low light, we'd get some kind of grainy image, just like you'd get with a photograph. If you had a very, very long exposure, you might get a blurry image, particularly if you're imaging something that's moving, like a human. And if you're imaging with a field of view of only 1 millimetre by 1 millimetre, you're especially sensitive to any kind of movement in the sample. And so you want to have something in the middle where you've got enough light that you can use an exposure time that's kind of between those two. Another key thing is that you can choose a single wavelength to do the imaging. So you can choose the wavelength that's optimal for your particular locations. If you've got a thicker sample, you want a higher energy X-ray in order to get enough photons through to be able to complete a measurement. But you don't want it to have too high an energy, or all the photons will go through and you won't get any kind of contrast. A single wavelength also means that we're better at measuring the phase of the wavelength of the wave field. So you can imagine if we've got lots of wavelengths present and the phase changes, it's hard to kind of differentiate one from the other. So just to show you here the spectrum, so this is which energies are present in the X-ray beam, So how many counts you get at each of these particular energies on the x-axis. So from the synchrotron, we get like nothing except for the energy we want, so say 20 keV X-rays. Whereas if we use some kind of conventional X-ray source, we get a spectrum that looks something like this. So a lot of different energies present. Most of these low energies here will just be absorbed by the patient or the sample, depositing dose without actually giving you any information. The high energies will probably go straight through. And if you've got a particular, so like tungsten will always have these peaks exactly here, no matter what you change on your X-ray setup, they're fixed. So the synchrotron allows us to really vary that easily and get a lot of light. The other key difference between these two is that this peak is way bigger. So there's a lot more light coming from the synchrotron. The other key advantage, if we want to use techniques that look at the phase of the X-rays, is that the synchrotron is highly coherent. So coherence, we get high coherence if we move far, far away from our X-ray source. So from these synchrotrons I showed before, the imaging beam lines where we do this kind of imaging are all the way out here, so 250 metres away from the main electron current, and this one's around 150 metres away, off in a separate building. And so this is just to show you the Australian synchrotron. All the experiments are kind of within this big building here. You might also see there's like a small synchrotron over here. That's just the barbecue shelter that they copied. And where we do the experiments is all the way out here. So we've got this long tunnel. And so this is just a video going down that tunnel. So you can see it's quite long. This is carrying the X-ray beam down the tunnel. We have to go down and press all these buttons to prove that we've checked and there's nobody in the tunnel before we can turn on the X-rays. So quite some distance. So at this large distance we get high coherence waves, and so as an analogy to kind of make that what coherence is more easily understood, I've got this video from a watering hole. It's at the bottom of a waterfall, so there's some waterfall landing down here. And you can see that it's creating waves going across the watering hole. If you're looking close to the source of the waves, the waterfall, you can see that there's waves, but they're all kind of appearing and disappearing. They're all kind of choppy. There's not really any nice wave fronts. But if you go way out here, you start to see these nice wave fronts that continue propagating. They don't suddenly disappear and appear. And they've got the same phase across some area. So the further we go from the source of the disturbance, the source of the waves, the higher coherence waves we get. So coherence is basically saying how uniform is the phase across the wave front. So kind of perpendicular to the direction of propagation, do we have uniform phase across there? And if we're in close to the waterfall, then we don't. If you're far away, then you do. So it's the same with the waves of light that we get from the synchrotron. If you're in close to the source of those waves, then it's quite choppy. We don't have this kind of uniform phase. But if you go a large distance from the source of the disturbance, so if you go all the way out here, then you do get these nice coherent wave fronts, so really uniform phase. And so that means we can see these variations in phase that are introduced by the sample. The other key thing that we can get by moving far, far away from a synchrotron source is that we get low penumbral blurring. So that basically means that we're not going to see the image kind of blurred out. So if you've got a very small source of light casting a shadow, you're going to get a nice sharp shadow. Whereas if you've got a larger source of light, where there's light coming from a larger area, you're going to get a blurrier shadow. And we call this kind of region around here the penumbra, which is where we've got this blurring around the outside of the object. So you can do that either with a very small source of light, or if you've got, if you're very distant from that source of light, you also see that effect. So we'll get less blurring of the image due to the size of the source if we go far away from it, and we're able to do that at the synchrotron, so that provides us with that high coherence and the low penumbral blurring. And the main reason that we can do that at a synchrotron is that the beam is, we say it's very collimated, so it's all kind of heading in the one direction. If we go to an X-ray source in the lab here, it's kind of spreading out in all directions. So if we go 250 metres away, we're not going to get very much light arriving there at all because it's spread out this kind of 1 on R squared relationship where we're going to see not much light arriving there, but because the synchrotron produces such a highly collimated beam, so all heading in the one direction, we can go 250 metres away and we've still got the whole beam that we can use. It hasn't kind of spread out in all directions. So going a long distance away gives us those 2 key advantages. Okay, so once we get inside the experiment, we've got this lead-lined room called a hutch. There's typically a shutter in the way here. So if we're taking images of something that's sensitive to radiation, like a living animal or even a very small sample, then we'll always stop the X-rays between every image. The camera at the back here is used, it converts the X-rays into visible light using a screen that fluoresces when the X-rays hit it. And then we typically have some kind of optical lens to focus in on that scintillator and capture an image using a CCD or a CMOS. The other things that we have kind of key up the front here is we've got, say, an undulator here creating the X-rays. We've got a monochromator, which means this light coming into the monochromator would have some range of wavelengths present, and then we just get one wavelength out the other side. So these kind of experiments, obviously we can't fit an entire synchrotron into every hospital if we wanted to do medical imaging. You can't fit it into every airport if you want to scan people's luggage. So the other key thing is moving to smaller X-ray sources. So in the last couple of years I've spent some time in Munich where they've got the world's first compact synchrotron. So you can see this one's much smaller. It's about 8 metres long instead of 1 kilometre across. And so it's much more feasible for these kind of widespread applications. So this one it works a little bit differently. We still have an electron beam coming in that's going to be accelerated, and then it goes into some kind of storage ring. But then instead of passing through magnets to make the electrons wiggle back and forth and produce X-rays, they are actually collided with a laser. So we have this kind of, the red dot there is the laser light. And so when it collides with an electron that's coming in at high speed, the electromagnetic radiation of that laser looks kind of like a whole lot of very small magnets with very, very small period compared to anything we can physically build, which means that the electron will oscillate back and forth within a very, very small space, which is what allows us to really make the whole system very compact. So this mechanism of the collision between the electron and the laser photons is called inverse Compton scattering, where you've got this high energy electron coming in and interacting with what's a relatively low energy photon. So the other way that it's sometimes explained is that the energy of the electron is given to the photon, which means that the photon goes from a laser energy up to an X-ray energy. So these are kind of the state of the art in terms of the imaging facilities. The X-rays that we get from a synchrotron are very high flux, monochromatic, highly coherent, and that means that we can do this fast imaging. We can do energy sensitive techniques, including phase contrast imaging, and we can get really high contrast using the phase information if we've got that high coherence. So some of the challenges in terms of using synchrotrons for this, the first one is the radiation dose. So in terms of putting humans in the synchrotron, it's not too widespread. There's been one study in Italy where they were doing some mammography screening using the synchrotron. But there was about 10 years kind of lead up of that, establishing the correct safety systems and everything. And then in the European synchrotron, they've done some radiation treatment for brain tumours. But that's really it worldwide, because you do have to be so careful because of the very high flux. There's potential for a very high radiation dose being delivered to patients. So you need good safety systems to make sure that the beam can be very quickly stopped. And the other thing that we can do to kind of move towards some kind of viable imaging at the synchrotron is to have very highly sensitive detectors. So we're really measuring all the X-ray photons that are coming out, not throwing away any of the information. The other thing that we can do is develop techniques that can use efficient detectors. So if we've got, for example, a detector with very big pixels, it's going to be much more efficient at collecting the light than something that's got very small pixels, so very small area for this interaction between the X-ray and the measurement. So that's the other challenge. And then, of course, moving these capabilities from the synchrotron to these more compact and low-cost X-ray sources. So I just wanted to show you one of the very fastest imaging that's been happening recently. So this work was done by Daniel Duke, who's in engineering here at Monash, and he was working with some people who wanted to look at how they should formulate their treatment that they put in a inhaler device, so like you might use if you've got asthma or something. And so in this particular study, they were capturing an image every time the electrons usually don't just kind of go around like a racetrack. They go around in little bunches around the synchrotron. And so in this study, every time the bunch came past, they took another image, which means there were 50 picosecond exposures, which was enough to capture this kind of sloshing of the liquid around. So they could study whether having some ethanol in their treatment was going to result in smaller kind of droplets of the treatment landing into the airways of the patients. And so this is probably the very fastest imaging that's been done anywhere in the world. And this one was done at the synchrotron in Chicago in the US. And so you can see here that you get these, around each of these air bubbles, there's kind of this black extra line, and that's from those phase effects. So if we just use normal attenuation-based imaging, you wouldn't really see anything. But because we've kind of added this extra distance, we get, just like we saw either side of the airway, there was those black lines. Around these air bubbles here, we see that kind of black-white fringe. So some of the applications where we can use phase contrast imaging is, of course, in medicine. So mammography is a big application, so imaging of the breast, because it's all soft tissue, so that's what you normally can't see with conventional X-rays, and it's where you can see something with phase contrast. There's biomedical research, which is what I'm primarily involved in, so trying to understand disease and develop new treatments. Materials and manufacturing, so it's been applied to look at different, let's see here we've got some kind of welding or something, I think, looking at how well that works. They've been using it to look at when they're constructing aircrafts, where it's most likely to fail, so you can see these kind of very small structures that could develop into cracks. And also in food quality control, so there's some markets that will pay a lot for perfect fruit. Sometimes it's hard to see if it's perfect from the outside, so they're willing to invest in these kind of X-ray devices to really test if the fruit has some kind of worm in it or something like that. So I'm just going to take you through one of the projects that I've been involved in and some of the applications that we've had of phase contrast imaging in that research. So the study is around cystic fibrosis, which is a genetic condition, and so that's seen in a number of ways, including digestion. It's very hard to put on weight, but most particularly in the airways. So they'll typically develop worse and worse lung conditions, so more and more disease, very vulnerable to inhaling any disease, which typically results in death early in adulthood. Although there has been very impressive jumps in the life expectancy. So I think in last year the life expectancy jumped from the early 40s to almost 50 just in 1 year alone. So it just shows how we're living in the right period if you want to get good medical treatment. The advances are quite impressive. So in cystic fibrosis, the main thing that's the problem is that the airway surface liquid is decreased. So all the airways in your body have this kind of liquid lining, which is there so that if you inhale any dust or debris, it sticks to that liquid. And then these little hairs that are in the liquid, called cilia, they kind of beat together. They're kind of beating like the legs on this little centipede. So here the centipede is moving the branch underneath him. In the airways, they're moving these little pieces of debris or inhaled dust or disease. They're moving that away from the lungs and towards the mouth. So this kind of coordinated movement of these little hairs pushes any of the inhaled debris away from the mouth. So with cystic fibrosis, that liquid depth is decreased, like we see here on the right, which means that you get a lot of debris just sticking there and not getting cleared properly, and so you get this build-up then of infection in the lungs. So most of the current treatments are very short-term. They're really just trying to slow the disease. And my collaborators at the Women's and Children's Hospital in Adelaide, they're looking at a longer-term treatment using gene therapy. So they had a few obstacles in trying to develop these new treatments. First of all, they didn't know if the treatment was reaching the correct site in the lungs. It's very hard to deliver to a specific part of the lungs. They didn't know if the treatment was working, and trying to get that kind of measurement quickly was a problem. So a lot of established studies would take an image of the lungs and then treat them, wait several months, take another image, and then compare the two images and try to figure out what had changed. And this is it takes a long time for one thing, and also it's not a very sensitive measure because it varies hugely depending on what else happened during those months. If the patient was living in a dry house, if they caught a cold during that time, there's a lot of other factors that can come in. So what my collaborators were really keen to see was this liquid layer increasing, this airway surface liquid increasing in depth in the seconds after treatment. If they could see that, then they would know that the treatment was really doing the job. The other problem was that it's difficult to detect small changes in health. So you're not going to suddenly go from having completely diseased lungs to completely healthy lungs and be able to measure that easily. So, for example, some studies you'd have to breathe through some kind of pipe, and you have to breathe air out as quickly as possible, breathe it in as quickly as possible, and they measure like a pressure flow kind of characteristic, which is measuring over the entire lungs at once. It's not measuring a certain part of the lung. So if you've just got one kind of 5% of the lungs that's diseased down here, and that's what you've treated, that's not going to really represent a lung. It's not going to. You're going to see that in the measurement that's measuring over the entire lungs so well. So to overcome these obstacles, we've done some imaging. So the first one was imaging treatment delivery, just where the treatment was going. We've measured how that inhaled debris is moved along the airway surface, how it's cleared. We've measured the depth of liquid that's in the airways, and also looked at local lung health measures. So most of these studies have been done in either a mouse or a rat. So this is a CT that we took of a rat at the Australian synchrotron. So this is with phase contrast, which is why we can see all the fur, and we can see the whiskers, and all that soft tissue, the lip there with all the pores in it, in so much detail. And so I think the phase contrast will kind of be taken away in a second, and then we'll see what it looks like just with the attenuation contrast that we would get from the bones. So this is just the bone sitting underneath, so that's what you might see in a normal X-ray. So with the phase contrast we get all that external and internal, all that extra soft tissue information. So we do still have the phase contrast here to capture the airways. So you see that main trachea there going down to the lungs that are shown in pink there. And we get, you can see those white rings of the cartilage that are around your airway to protect it. And you can see all those little tiny air sacs in the lungs there, which again we only can really see those because of the phase contrast information. We can also go inside those airways and go down through that 3D volume. So this is where any kind of treatment might go down here, and any inhaled debris should go the opposite direction. So this is the lungs here where the airways split into two, and then we see lots of airways going off in different directions to all different parts of the lungs where we've got these little air sacs to collect the oxygen. So the first thing was treatment delivery. So we've taken image sequences looking at treatment that's administered through the nose, and we can look at different volumes of liquid to say which part, how much liquid should we put in and how quickly to get it to different sites within the airways, if we just want it within the nose or off to the side there. So we can determine how much volume is required. We can then correlate how much volume we needed to put in to get a certain effect from treatment at a particular site. And a lot of these treatments that they're delivering are many thousands per microlitre, so it really, we don't want to be just pouring it in there for no reason, so you can really optimise how to deliver these very expensive treatments. We've also looked at treatments into the lungs. So you can see here we can try to target one particular lobe of the lungs that we see here, or we can deliver the treatment all the way across the lungs. The second thing we looked at was the clearance of the debris along the airways. So if we zoom into the trachea, so this is the kind of lying on its back, we've got the head and the lungs and this main airway coming up here. If we zoom into that trachea surface, we get that strong line at the top and bottom of the airway. And we've also put in some inhaled debris there. And so we can look at how that inhaled debris is cleared when we've got either no treatment, some kind of control treatment, so some liquid that we don't expect will have any effect, but we'll put in the same volume as we do of the active treatment, which is the third option there. And so we see that if we've got some kind of active treatment, we get a lot more clearance of the inhaled debris. But really any hydration of the airway surface does help in clearing that debris. And so we've done that in the trachea, we've also done that in the nose. So there's particular airway surfaces in the nose for cystic fibrosis that are affected more or less than others. So we can see how well these newly developed treatments work. You can see here the active treatment is really clearing a lot of particles on the right there due to the mannitol. And we've also done it in the lower airways down towards the lungs. So this was actually captured using that compact synchrotron in Munich. So these little dots is the inhaled clearance, and you can see also the type of behaviour. So these ones are kind of in a little train here. You see them grouping together, kind of collecting their friends here into what we'd call a mucus plaque. So they collect into this little group, which makes them clear more easily. So we can look at, can treatments also kind of aid in those kind of processes? The third thing we were looking at is regional lung health, so looking at the motion of the lungs. So Steve Dubsky, who's your normal lecturer for this, he does a lot of work in this area. And so using the phase contrast, you can see in this image here that the lungs give you this kind of speckly pattern. So that's because all the air sacs in the lungs are acting kind of like small X-ray lenses to focus the light into all these little bright dots. So we get all these little white dots here. And so if you can track how those dots move during the breath cycle, then you can actually track how each part of the lung moves during the breath cycle. And so that's used, this has been done using these PIV techniques, tracking those speckles in 3D, because we've actually collected data sets where we've collected images at all the different angles during the breath and reconstructed them into a 3D volume. So then you can see if there's a certain part of the lungs which isn't inflating in the way that it should, and see if that changes as a response to treatment. So we've applied this to look at cystic fibrosis mice and looked at how these different measures change in the presence of the disease that we get with cystic fibrosis. So these, in the plot down here, we've got the blue dots are the mice that don't have cystic fibrosis, but they're litter mates, so they've got the same mother and they've kind of grown up in the same environment, so they're like the control. And we see that they're really grouped quite separately in terms of the resistance of the airways to motion. And we can also categorise the type of disease. So if we look at this particular mouse up here, then we can see that one half of the lungs is not inflating in the same way as the other half, whereas over here we can see that there's much more even inflation. So you can really isolate where the lungs are not healthy and then try to correct that. The last one that we were trying to measure was the depth of the liquid in the airways. So the cystic fibrosis, we see that liquid depth is decreased, and effective treatment should increase the depth of that liquid so that we can clear any debris that we inhale. So we tried to take a propagation-based image of the side of the airway. We got these kind of images. So on the left is in vivo, so that's just inside a living mouse. On the right we have ex vivo, so this is just the airway that's been surgically removed, and it's just sitting in a dish of liquid. And you can see the edge of the airway very clearly, so we've definitely got air on this side and kind of tissue on this side. We can see these speckly bits are the cartilage rings that protect the airway. But we don't really see any difference between the airway surface liquid, which is kind of a salty water, and the tissue, because tissue is mainly liquid, so they're very, very similar, so it's hard to get any contrast between the two. So neither in vivo nor ex vivo could we see the other side of the airway surface liquid. We could only see the liquid to air side. So propagation-based imaging was not sensitive enough to reveal the airway surface liquid. It has the other kind of key advantages that we needed in this application, in that it uses a very short exposure time. So when we're imaging the surface of an airway, you can see we're at quite high resolution. This is a 100 micron scale bar there. So every time you breathe in, the airway surface usually moves by around 100 microns. So we need to be able to do a really short exposure to capture that, a sharp image without motion blur. And it's also quite high resolution. So propagation-based had these advantages, but not enough. We then tried a method called analyzer crystal-based, where the wave that's come out of the mouse is then bounced off another crystal. And you'll see that the amount of light that's bounced off the crystal depends on the angle that it's coming into the crystal. So if it's coming at the right angle, you'll see a lot of light reflected, like you do at the side of the airway here. Whereas if it's coming in at the wrong angle, you won't see much light reflected as kind of over here. And so this was kind of promising in that we could then see, I don't know how well you can see it there, but there is this other interface kind of down there. Can you see a line? Maybe you have to like squint a bit, like magic eye or something, but it's, we could actually see the difference between the liquid here and the tissue that was sitting here. So we could then measure the airway surface liquid. The problem was that this was taken with an ex vivo airway, so outside the body, surgically removed, with a 3-second exposure time, which is impossible if you're going to have any kind of living animal. You're just going to get a big blurry image if you take an image for 3 seconds. So this was not sufficiently fast enough to capture this information. So we had to come up with some method that would meet all three of these. And so the answer we came up with was what we call single grid phase contrast X-ray imaging. So if we're talking about capturing soft tissue structures, we're talking about soft tissue is where the light goes through, but it will change direction. So we do see these changes in phase. So as a visible light analogy, the water that you would see in a swimming pool, the light goes straight through it, but it does change direction. And so the challenge that we're addressing is kind of like the same challenge as me asking you, can you figure out the shape of the water in this swimming pool from this image? So there's two ways you could do that. The first would be to look at all these white lines that we see on the bottom of the swimming pool, where we've effectively got lenses sitting on top of the water, in that each of the waves in the water acts like a lens to focus the light and create these white patterns. So we could kind of go backwards from those to figure out what the surface of the water looks like. Is there any other way? What else could we use? If you want to work out the surface, what the surface looks like from this image. I'll zoom in a bit to kind of help. Yeah, exactly, the tiles on the bottom of the swimming pool. So you can see that the tiles here are distorted according to the shape of the water that's in there. So it's kind of acting like a magnifying glass right here and magnifying those particular tiles. These ones over here are kind of demagnified and squashed together. And so we wanted to use this same kind of property to do phase contrast imaging with X-rays. So what we did, we'll start off with the diagram. So if we've got kind of a cross-section through the pool here, we've got the tiles on the bottom of the swimming pool, we've got the water that's in there, there's some waves in that water, and then the air above that. So if we imagine the light that's coming from the bottom of the swimming pool through the water and up towards your eyes, if we say that it has uniform phase in the water, we'd see some kind of variations in the phase up above the water here. And so we could also trace the light that's kind of coming up from each of those grout lines to see that some of them are going to be kind of squeezed together and some of them are going to be spaced out, depending on that surface of the water there. So if we then want to go from this to X-rays, I'm going to turn it sideways because usually our X-ray sources project light across, not up. If we go with X-rays, we're going to say we're going to be looking at soft tissue instead of water. If we're using X-rays, we're going to see that the angles that the light changes by are much, much smaller than you would see with visible light. So we're going to need much larger distances before we see the same kind of amount of squashing of those two lines there. Do you want me to close that? Is that noisy or is it you can still hear me? Yeah. The other key thing with X-rays is that they bend in the opposite direction to visible light. So we'll kind of see that these beams are going in the opposite direction. But apart from that, it's basically the same setup. We've still got this light going through, some phase variations coming out the other side. So this is the setup. We use a grid to produce a reference pattern. We've got a camera at the other end, and we've got some kind of X-rays from the synchrotron, which are coming through, and we're going to see these variations in the phase. And so this should mean that the further we go from the sample and the grid, the more and more distorted that grid pattern will be. So we tested this using some plastic spheres. Plastic has very similar X-ray properties as muscle and fat, so that's quite convenient for doing testing. So here there's a big sphere down the bottom here, and then there's like another sphere there. When I say big, they're about 1.5 millimetres each. But you can see that they are distorting that grid pattern, just like you see the water in the swimming pool is distorting the grid pattern of the tiles on the bottom. So we see between the spheres, there's not any movement of it. It's kind of like an empty swimming pool. But where we've got a really kind of steep gradient in the thickness of the spheres, we see that it kind of pulls that grid pattern across here, kind of pulls it down here, and kind of pulls it diagonally here. And then when we get towards the middle of the sphere where it's quite flat and the thickness isn't changing very much, then we see that there's not so much a distortion. So we then have to somehow quantify that. We can definitely see that there's some spheres there. To quantify it, we do this locally by looking at these two images, extracting a tiny region from each of them, and then saying how far has this particular part of the image been moved when we introduce those spheres. So we do that using a cross correlation, which is effectively sliding this window back and forth here to extract how far that part of the image has moved. And then that will give us a vector shift S, which will have some shift in the X direction and some shift in the Y direction. So this part of the grid here is mainly a shift in the Y direction. This one's mainly a shift in the X direction over here. And so we can get those two parts out and then do that at every single position across the image. So we do take these small windows out from each position and scan all the way through each pixel in the image to get something like this. So this is showing you how far that grid pattern has been shifted by those spheres. You can see it's not very big, it's just a few microns, but it's enough that we can see something. So we see, of course, that big shift across in the X direction, in the negative X direction there, and the positive X direction here. We see the Y direction that we get the biggest shifts here and here. And of course, there's about zero shift in between those spheres. We can also take information about those images. So if we take those shift images in microns of shift, then we can use the geometry of the whole situation, so that propagation distance Z, and then do an inverse tan of that. So we've got kind of a big triangle there. You guys don't need to know all this, by the way, it's just so you can kind of get an idea of what's involved, which can then give you a phase gradient in radians per micrometre, which we can then also integrate together using a Fourier transform and the Fourier derivative theorem. There's some automatic timing going on now, it's doing it without me, to get the total thickness of the sample there. And so if we look at that, we can actually see we get a very accurate quantitative reconstruction. So if you trace this blue dotted line across the image, you can see that the total thickness kind of drops to 0 and goes back up again here as we move across that dotted line. And the black line here shows you the theoretical thickness of 2 perfect spheres. Obviously our spheres aren't perfect, they've got some little scratches on them and things, and we get these light blue reconstructed thickness, so we can really get quantitative measures. So this, again, you don't need to know this equation, just to kind of show you where some of this contrast comes from and some of the theory behind it. So this is what we call the transportive intensity equation, and it describes how the intensity of this wave field that's propagating from some sample to some image is going to change as it moves with propagation distance z here. K just describes the wave number, so which describes what wavelength light we've got coming through. But basically if we have z equals 0, this whole second term equals 0, and we just get that the image at the image plane here is the same as the image at the contact plane, so that kind of makes sense. But if we've got z not equal to 0, and we have some phase gradient, so this here is taking the derivative of phi here, the phase. If either this term or this term is non-zero, there's some phase properties that are in the wave field coming out. We're going to see that there's a difference between the image in the image plane and the image in the contact plane. There's some difference here, which increases with propagation distance and also increases if we've got bigger phase gradients. So this first term here is where we see a lot of the propagation-based phase contrast. So if you think about this cylinder here, the biggest gradients in the phase are at the edges, so where we're kind of stepping up onto the cylinder is where it's steepest. So that's where we see a lot of the contrast. This bright dark fringe is right on the edges. Whereas with this grid-based methods, the most of that contrast corresponds to this second term here, where we've got, we've introduced a derivative in a large intensity gradient here, and that's a grid line. So that's a high gradient in intensity going from having a lot of light that goes through the holes of the grid to very little light, where we've got the shadow of one of the grid lines. If we introduce that, then we can reveal this phase gradient. And if we just take the gradient of the cylinder, you can see we get information kind of going all the way in towards the middle of the cylinder, which means that we're a lot more sensitive. So if we had no intensity gradient here, this whole term would be 0 and we wouldn't see anything. So if we had painted the whole bottom of the swimming pool the same colour, so we've got the same intensity light coming from it, you're not going to see distortions to the tiles because you don't see any tiles because it's all uniform. So it's basically we need to have some kind of pattern on the bottom of the swimming pool in order to do this kind of reconstruction. And of course the moving water is a good analogy because most of the samples that we image are also moving. So we don't actually have to have these perfect tiles on the bottom of the swimming pool. We could have some stones on the bottom and we would still see the same kind of behaviour, right? You can still see, if you look at these stones here, that they're kind of wobbling about according to the shape of the water that's above them. So one of the advances that we made was we don't actually need to have this perfect periodic grid pattern. We can use any kind of pattern. So for example, the speckle that you would see if you put a piece of sandpaper in the beam. So it's this kind of random pattern, but it still shows you where the spheres are. You can see those same spheres in the same positions. We see this kind of movement of the reference pattern, but it doesn't need to be a perfect grid. And so this is quite useful because we can get the same kind of images out, but sandpaper is much cheaper than a grid, and also it is, we can get much larger areas for doing large-scale imaging very easily, whereas making some of these grids perfect over a large region is sometimes difficult. So we'll come back to, we wanted to measure this airway surface liquid. Does this method work for that? Indeed, it is sensitive enough. We can see with this grid method, this second line here between the liquid and the tissue, and then this line here between the liquid and the air is very, very strong, so we can measure that airway surface liquid. It uses just a single short exposure, so that's fine. It's no different to propagation-based imaging in that regard. The resolution, you could say here, this image does not look as beautiful as this image. It's kind of got these blocky parts that are kind of left from the grid pattern. So what we can do if we want to get a really beautiful image is we can use what we call a phase grating. So phase grating is kind of like a whole lot of X-ray lenses. So the light goes from the top down to the bottom through here. And if it goes through one of these gaps here, it's going to come out completely out of phase with the light that goes through one of these pillars here. So a shift of pi between them. And if you go to the right distance beyond that, then you're going to see that you get a grid pattern something like this. So instead of blocking some of the light to create a grid pattern, we're kind of redirecting some of the light, focusing it into a grid pattern, which means that we're making use of all that light. So we can capture two images. This is the grid. It's very hard to see because it's so fine. And then the head of an ant. And we can look at how that grid pattern is moved horizontally to get this pattern and vertically to get this image. They look very similar, but it is useful to have both of them. Sorry, it's advancing without me again. It's useful to have both of them because there's some features that you see in one but not the other. So this vertical airway here, you don't see in this image so strongly. These little horizontal hairs here, you don't see in this image so strongly. So by capturing both directions, then you're sensitive to all the features in the image, and you can then integrate them together to get the total thickness. If you've got a very fine grid, you can also just get rid of the grid to get what's a propagation-based image, so it has this bright dark fringes around the outside. And you can also look at how the grid's blurred out to give you information about sub-pixel structures. So we can use this method to image anything really small at high speed. So this is a fly that landed in the beam. And you can see again the advantage of having both of these. So like this little spike here you don't see in this image. These little fang things you don't see in this image. So it's useful to have both of those at once. And then you can also extract these ones down the bottom. So you might have noticed that the beam was kind of flickering, that it looks like kind of low quality, and you're like, oh, it's not so nice. The reason is it's actually so sensitive that when the light's bouncing off this monochromator crystal to get the one wavelength of light, that crystal is just kind of oscillating ever so slightly, and when you're 250 metres away, the wavefront is kind of coming in like this. It's not always exactly the same angle, and this imaging technique is so sensitive that we can even see those effects. So it's actually kind of showing how sensitive the technique is rather than being bad. Yep. He didn't die, but if you do it for a long time, he could. Yeah. There's no ethics guidelines around imaging insects. The ethics boards don't seem to care about them. Yeah. I'm sorry. I hope it didn't pain anybody. He's okay. It didn't burst into flames. I haven't seen anything burst into flames. I've seen the most radiation damage I've seen is when I had a clear perspex cube that I was using to refract the beam, and I just kind of left it in the beam all day while we were doing experiments, and it kind of had this brown line burned through it. Like it still looked alright, but it looked like the beam had been like captured inside there. There was this is exactly where the light had gone. Yeah. But there's for doing any of this animal imaging, there's like very strict guidelines about what we're allowed to do and what we're not allowed to do, so that like the mice are never in any kind of suffering. They're always asleep when they're being imaged, and they have this board of people who have representatives from the community, from animal aid kind of organisations, from vets, from researchers, and they review anything that you ever want to do to animals. So, yeah. If there was an alternative, we would use it. But there's not yet a kind of synthetic thing that we can have responding to an airway treatment. So, yeah. Yeah? To large-scale applications?
For example,
What was the last bit, sorry?
the endobacterial pipes or something.
So like imaging, can we image large objects? Yeah. So the challenge is that we need to be able to see these phase effects. So like as we saw before, that grid pattern moved by like 2.4 microns. So if you're going to image an area that's like a metre across and still see these very small changes, you need like custom detectors that have many, many small pixels so that you can both see the small changes and capture the large area. So there kind of needs to be some larger adoption so that companies start to make these detectors. Or we can use other techniques where we place, for example, a second grating afterwards which amplifies the signal. So, have you ever seen like, if you've got like a net curtain kind of moving past itself, sometimes you see these kind of bright dark bands. Or if you take like a video or a photo of somebody that's got like a stripy shirt on, sometimes you can't see the stripes. If they're really, really fine in the picture, they might kind of look like these really strong bands, these Moira artifacts. Anyway, watch out for those, because that's something you can do which makes these small shifts into something that's bigger, that we can then measure those at the same time as measuring a big object. So that's definitely one of the challenges. Yes. Yeah, and that's the other possibility. So like most fluorescence imaging that's done at the synchrotron, that's what they do. They have a very small beam and they move it across and look at how each point fluoresces different wavelengths. The difficulty is the speed, I guess. So trying to image like this movie by doing a raster scan would be impossible because these are like 15 millisecond exposures. So we don't have anything that can move that quickly and consistently to do it. But that's the other possibility if you can get, yeah. And hospitals, like in terms of medical imaging, hospitals very much want fast techniques as they don't want to spend many millions on something and then it only does one patient a day or something, so, yeah. Cool. So anyway, so we've kind of met this last tick now in that we can do this higher resolution imaging. We can also do imaging, this isn't quite as high resolution, but where we just put the grid across half the image. So this is inside an airway here. We've just got the grid on this top part here, and then we've just got propagation-based imaging happening in the other half. So we can see this airway surface liquid here, and we can also see these beads moving here or here with kind of either modality. So this has allowed us to measure these time sequences of the liquid. We've did that first in these ex vivo airways, and we saw that with time on the x-axis here, that the depth of the airway surface liquid increased the most in response to the hypertonic saline, which is kind of a known treatment, compared to if we used these kind of sham treatment in the red or no treatment at all in the green. We also saw that this tissue here kind of squeezes, so the liquid comes out of this tissue like squeezing a sponge, and you see that the tissue thickness actually drops when we have this hypertonic saline as this part here squeezes the liquid into the airway surface here. We also did that in vivo, so this is again with the big grid, so it's not so beautiful, inside the living mouse, with this new treatment HSP308, which they're developing in the US, and we can see that that depth of the airway surface liquid stayed up at this increased level, so that's quite promising. Okay, so just to summarise, so phase contrast X-ray imaging can really help us in medical studies in that it reveals not only the structure of the body but also the biological processes like how the lungs breathe, how the treatment will affect the airways, and so we can see these changes in real time. So there's a few different methods to convert the phase information into something that we can measure, so some kind of intensity variations, so there's the propagation-based, and then this single grid method as well. So this has helped us in the cystic fibrosis study in that we can see how the treatments are delivered, how those particles are cleared along the airway surface, we can see the changes in the depth of the liquid, and we can also see local measures of the health of the lungs. So that's it. If you've got any questions, I'd be very happy to answer. Any more questions? Yep. Yeah. Yep. It was back a little bit. Yep, there it was. Yep. Yeah. So MRI usually takes a couple seconds to collect an image because you've got to wait for all the magnetic spins to go one direction and then have them go back down again. And so these images were taken with like 50 picoseconds or 3.7 microseconds between each image. So if you did an MRI image, you would kind of be like averaging several hundred of these images all together. So it would just be kind of one big blur. You wouldn't be able to see, okay, here's the bubble moving across here and now it's here and now it's here. Yeah. So it's the fast speed that's really useful. MRI, I'm not sure how good MRI is at seeing. It's like you get some kind of signal, but the other difficulty is you might not be able to resolve these directly. So MRI has a resolution more like a millimetre or something, whereas with this X-ray imaging we go down to the micron level. So it may be that this would be like one MRI pixel and this would be another MRI pixel, and so you don't see much detail. Yeah. Anyone else? Yeah.
Could you go over how you created that 3D image of the mouse?
Yeah.
I kind of get how you could take it from different angles, but
Yeah.
also getting the depth into the mouse.
Yeah. So a normal MRI scanner, you've got like an X-ray beam that's like a line, and then you've got a detector that's a line, and so then you move that around and you collect these projections at all the different angles and reconstruct one slice. So that's conventional with an MRI scanner. With a CT scanner. So with the camera we use here, it's like a 2D array. So we're effectively collecting a whole lot of CT slice. Each line of pixels in the X-ray detector is giving you one slice worth of information once you put it all the way around. So we get like one slice through here, and then the next row of pixels is like the next slice through here, and then the next slice through here. And by having many stacks of slices, or one big stack of slices, then you get this 3D volume. So when we're so we basically take a whole lot of images all the way around, and then we say, okay, I want to take row like 50 from the image, so go up 50 pixels, take that row from all those images, and then we say, okay, we know the projected, gives you like a projected thickness multiplied by density image. And so you can use what's called the radon transform, which goes from like an object to the projected thickness times density. And you can get all of these pieces of information and kind of fill in this radon space, and then do the inverse radon transform to go back to the total kind of density, because you've got thickness times density and then you know the total thickness of all of them added together, so you can go backwards to get the density, which is how you kind of separate out one object from another. Yeah. A minimum thickness? The slice, the smallest pixel size, I guess. So it's probably around half a micron with the current technology. Yeah. Yeah. Anyone else? Does that mean we're finished? Yep. Okay. You're free to go. Oh, thank you. Feel free to come and ask me questions after if you like. I'll be here.
Correcting the endotracheal tube. We are looking for errors where the tube goes into the esophagus, or it inserts itself into one particular lung, the right lung supposed to the left lung.
Okay.
So we're looking at optimized, we actually do economical imaging solutions. Yeah. So we rounded out that, we've come to a conclusion that EIT is good because and we're looking at emergency services, integration of emergency services, so we can't have a CT or an MRI closer. So we're looking at EIT for now. So do you do any technologies which would serve our purpose, which would be economical as well as portable.
Use it for a larger scale.
Yeah.
Then we use this for an instant image.
For an instant image which is portable, which
The portable bit is the difficulty, I think. Like there are portable X-ray sources that they use. Like if somebody leaves a backpack somewhere and they want to see if there's a bomb in it, they'll take that. They've got these little robots with X-ray sources that they can take images. But I don't know if you'd be sensitive enough to like that you could use that source to do phase contrast X-ray imaging to
That
see
won't
the
be
airway.
sensitive enough to send some tube or something.
We just need to see the distribution of air and also the positioning of the tube. That's it. So
Yeah. It's probably not there quite yet, I would say, with phase contrast X-ray imaging. Yeah.
So
Yeah,
how big is
it-
your, like, how big is your
The X-ray
phase
source.
contrast X-ray.
Usually, so the Australian synchrotron, which is the biggest one, the beam is like this. So it's about 40 centimetres by 3 centimetres.
so it's a portable.
No, that's the entire synchrotron. So you have to take the
Oh,
synchrotron.
that photo, yeah, that photo.
Yeah.
Oh, okay.
I
I thought
just
that's
see the setup of the Beamline 20 XU,
Yeah.
and it seemed pretty big. It almost occupied a.
Yeah. It's like a whole.
It's a whole room. So
Yeah.
I'm wondering if something smaller has come out.
It
Something that
hasn't.
has it, is it?
Not yet.
It's the closest. We came to the conclusion that EIT by Draga, the Palmo Vista,
Okay.
that was okay. That was the closest, which is portable, and you have a belt around which, and then you have a monitor. That was the closest
Ah, cool.
portable thing we could come across.
Yeah.
I'm wondering if there's any new technology we could work on. But then
Yeah.
face contrast is too big.
Yeah.
MRI is
It
too big.
is. Maybe
CT
in
is too
10
big.
years, come back and
Yeah, we thought the photo you were showing,
Yeah.
it's like the entire lab or something.
Yeah.
It's so.
Yeah, it is. And like the synchrotron is like a kilometre around. It's like,
Way
yeah.
too big. We need
It's like the
something-
opposite of portable.
We
Yeah.
need something which should probably filter now.
Yeah.
It's fine.
Yeah.
Thank you.
Sounds cool. Good luck.